We opened the meeting talking about the value of perception, and I thought I'd give you an idea of the vantage point I bring in terms of the slides I'm going to walk through. I spent a reasonable amount of time not only in drug development companies and small and large pharmaceutical companies, but also in small and large in vitro diagnostic kit companies, and then lastly in small and large CLIA laboratory developed test service laboratories. So I kind of stitched together that experience in general. And then a little bit closer to home, I represent kind of a downstream stakeholder in NASH. And the reason for that is right now I oversee the science at CareDx, which is a transplant diagnostics company near South San Francisco. And as you might expect, there's lots of discussion about the not too distant future when NASH will eclipse HCV as the primary reason for liver transplants. And my concern is twofold in that regard. If we don't intervene, there'll be a lot more people that we need to deal with. But at the same time, it is highly likely that the pathophysiology of disease doesn't have the cornerstone solely in the liver. So when we transplant livers, we don't know if we're transplanting a liver which in fact comes from somebody who's predisposed for NASH, or that the recipient brings a predilection to liver damage. So we both have to be concerned from the standpoint of transplant, and we have to be concerned from the standpoint of ongoing NASH once the transplant occurs. And then lastly, we share a lot of aspects in terms of how we think about managing the patients each of us see. We also use biopsies in kidney and heart, and because we don't have very many tools, and the biopsies that we use are as compromised as they are for liver for NASH. And then lastly, there is a real paucity of markers. So what I thought I'd do today is I really benefited from being here since yesterday, and although these are my core group of slides, I've taken the opportunity to modify some of those as well as subtract and add some of those. What's convenient is that some of the slides you have seen before, and although I will give you my vantage point, I think we can go through them relatively quickly. This is my disclosure. This represents some of the points I'm going to walk through in my talk. And this represents what I consider to be a functional definition of a diagnostic test. It's the translation of the complex and fuzzy spectrum of pathophysiology of disease into practical, actionable next steps for clinicians and patients to improve patient outcomes. Very simple. So one of the questions you can talk about is context. So have there been changes in clinical diagnostics over the last 5 to 10 years? And indeed, there have been many. First, there's an increased complexity of our understanding of disease. Breast cancer used to be one cancer. Now it's at least 5 different cancers, because we've begun, with the use of biomarkers, to understand the underlying etiologies and how they present. There's been a formal phased development of diagnostic tests similar to drugs. In fact, they share the fact that there are 3 of them by coincidence. I'll be spending a little bit of time talking about that, but at least we have a formal consensus development process to go forward. And a high quality of evidence is needed to be provided by the test service in the context of CLIA and test kit providers. It used to be that in the IVD or the CLIA world, you can kind of throw a biomarker out there and people would try to find out what the indication was that it was good for. That no longer is true, and it's now up to the service providers and the kit providers to in fact gather the necessary evidence, not unlike drugs, the necessary evidence to convince regulatory authorities as well as payers that they should be reinforced and adopted in general. It used to be that clinical validity was sufficient I'll be talking a little bit about that. The three phases are analytical validity, and clinical utility. It used to be that payers were quite happy to reimburse with clinical validity. That's no longer true. Clinical utility is required. And in brief, you can think about clinical utility as the demonstration that not only is a biomarker different between those with disease and those without disease, but in fact, by intervening, by using that piece of information, you can alter the outcomes going forward. As I thought about the presentations, I couldn't help but think that we tend to conflate two paths that sometimes are in parallel, but sometimes cross. And I refer to those paths as a biomarker discovery, which can be thought of as an exploratory walk, and the translation of a biomarker for clinical practice. And I think of that as a directed path. So let me just take a minute to describe for you what I consider to be the features of each one of those approaches. For biomarker discovery, you're really biomarker-centric. We talked a little bit about that yesterday. You think about the biomarker. It promises key insights into fundamental underlying pathophysiology. There's a plethora of biologically plausible biomarkers. There is more biomarkers, arguably, than we know what to do with. It benefits from a deep understanding of biology and how to take that forward. And the correlations that we look at really can take advantage of what I consider to be group diagnostic metrics, like receiver-operator curves, like sensitivity and specificity. On the other hand, the translation of a biomarker is fundamentally different. That directed path has to start, and I had to smile. The organizers are to be applauded for allowing the talks from last year and the webcast to be available. And Arun, I'm not sure if he remembers, he began his presentation by saying, "I'm just a simple doc." That's certainly not true. But his point was that you start with the question that you want to answer, because that's where you want to be. And that is the element of translating a marker. You need to start at the end of the road and then work back. It promises to improve patient management. There are actually very few markers that are developed to the point where it actually makes sense to go forward in that regard. And it really benefits from translation and diagnostics development paths. And then lastly, it really requires not ROC curves, not sensitivity and specificity, which are elements of the test, but what you need is information about the patient, the subject that's sitting across the desk from you. And that requires predictive values, that requires Bayesian statistics. The next slide is a slide that I don't want to spend a lot of time with, but suffice it to say, I think of assays in two categories, particularly in the context of those pathways. I think of them as clinical grade assays and research grade assays. The group at CareDx recently published a paper on the analytical validation of a next-gen sequencing assay, where we're able to measure the damage, the ongoing damage to an allograft in a kidney, a heart, and a lung in an ongoing basis. And this table walks you through some of the elements that distinguish a research assay from a clinical assay. And let me make a couple points. There will be times when people talk to you about a biomarker being validated. I don't think that's true. I think what gets validated is a biomarker assay, because assays perform differently. You don't have to look any further than breast cancer and HER2 assays to appreciate that they come in different colors, and some are useful and some are not. So it's really important for us to think about the performance of an assay rather than the performance of a marker. Clinical grade assays are, I had to laugh. I was talking to a friend of mine, and he said, "Yeah, I have a clinical grade assay. I ran some clinical samples." It takes more than that, and as mentioned by a couple individuals from the FDA, it takes rigorous analytical validation. And I would point you to the papers that we published at the end of last year to give you an idea, at least in the NGS context, what that means. And then clinical grade assays have to be of the highest quality, because now you're making patient decisions that may not always be life or death, but they're critically important to outcomes for patients. I don't want to spend a lot of time on this slide talking about the statistical or diagnostic metrics for test performance, but let me make a couple points. One is that there's no magic single statistical measure which gives you all the information you need. You really need to bolt together multiple pieces of information. I've already indicated that predictive values are absolutely critical from the context of a clinical grade assay. ROC curves, and we'll return to this momentarily, they have a qualified contribution. The reclassification statistics that people have begun developing to ask, does a patient move left or right relative to a threshold with an adjunctive test, I think are key pieces of information. Bayesian models are important. And then lastly, one of the nice things about the fatty liver disease is that, as Arun just mentioned, cardiovascular disease is among the comorbidities, and there's a pretty mature role of diagnostics when they play in cardiovascular disease, and we can use those from the standpoint of kind of best in class. This slide allows me to remind you that although ROC curves are nice from the standpoint of being a threshold-independent technique to visualize dichotomous diagnostic test performance, they really have significant drawbacks. So let me mention a few of them. First of all, the ROC curve basically plots false positives versus true positives, but it does that for every possible cutoff, including regions that aren't clinically relevant. So the number may not be helpful for us. It requires a highly accurate and related reference method to be informative. Let me just take a second. Arun and I were talking a few minutes ago. One of my pet peeves is people talk about gold standards, and then usually they follow that with it's not gold and it's not a standard. Well, then let's not use that term any longer. Let's use reference method, and we can appreciate that there are flaws in the comparisons that we're doing. In addition to that, when you look at AUC, a test that has high sensitivity or high specificity will appear to perform the same. False positives and false negatives are weighted the same, though, as you might expect, they have different trajectories and costs and consequences. So we need to think about which direction a patient goes, if you will, importantly. It doesn't address predictive values. We've already talked about that. And it's also actually insensitive to changes in absolute risk. I'm not going to spend a lot of time here because I think most people have a pretty firm understanding of diagnostic metrics. But I did want to highlight again that sensitivity and specificity is a measure of the test, whereas predictive values are a measure of the subject. And the really important aspect of predictive values is they are driven by the prevalence of the population that you're testing in. Sensitivity and specificity doesn't change, but predictive values change. And I'll show you a table in a little bit. And it's really important, and I can't help but say when anyone presents negative and positive predictive values, I almost have to say, but what population, what is the prevalence, and when you did your test population and your validation population, where the prevalence is the same. So these are really important questions that we need to ask ourselves, and that not only applies to discovery sets and validation sets, but also in the context of how we use the test. If there's a specific prevalence with a specific positive predictive value in your experimental studies, and then you moved and you go to the FDA and you're now applying it to a group that has a different prevalence, there could be remarkable differences. So how remarkable can those differences be? This table summarizes those for you. A test, and this is a hypothetical, with 95% sensitivity, 95% specificity, which probably outforms any test that we've worked on, can change from a probability of having the disease from 1.9% with a prevalence of 0.1% all the way up to 95% if the prevalence were 50%. So the test doesn't change, only the prevalence is changing. But you're looking across the table and giving fundamentally different information to the patient that you're speaking with. So the context of use is critically important. And although there are some people who would love to believe that high negative and positive predictive value are required for a diagnostic test, there are times when very low positive predictive value is coupled with very high negative predictive value, and it's the high negative predictive value that brings you the value in the context of use. The next slide reminds us of the hierarchy of evidence. Again, this is a familiar slide to many of you. I just wanted to use it to point out two things. One is when you look at a study, rarely do people spend the time to describe for you, was that a prospective study or a retrospective study? Were they opportunistic samples, or were they collected over time? Was it from a single center or a multiple center? And then was it an observational study or an interventional study? One of the most interesting things before I leave this slide that I'm particularly excited about is the use of registries, real-world data, and electronic medical records. They're not going to have the value of randomized controlled trials, but they bring incredible value because we know that some of these RTTs aren't very generalizable. So we need to begin stitching together the evidence that we have, and I'll be commenting on that. Michael Laure was at the Cleveland Clinic, now at NHLBI. This gives you an idea of how fast things can change. In his Ansel Keys Memorial Lecture in 2008, published in 2009, he basically said, if it's not a randomized controlled trial, it's not evidence. In 2013, you can see another paper by Michael, appeared in the New England Journal of Medicine, the randomized registry trial, the next disruptive technology. Things are changing. The National Academy of Medicine, used to be the Institute of Medicine, put out a report about 4 or 5 years ago about rapid learning, the ability to use electronic medical records, particularly in the oncology space, but we can use it in other areas. And then there are sentinel registries being put in place by NIH and other organizations that are going to be critically important. These are the 3 steps that we talked about: analytical validity, clinical utility. You don't demonstrate analytical validity after you demonstrate clinical validity. That's part and parcel. You can't change the assay once it's locked going forward. I don't need to spend much time on this. Robert did a nice job, and other speakers as well. It's referred to as the context of use. It comes back to Arun's point of what's the question you're asking, because unless you know what the question is, you don't know what kind of assay you're building. You don't know how it's going to perform. Do you want to exclude patients, or do you want to include patients? The one thing I might note is that we talked a little bit earlier about, so what's the intended use? Because that's the key question that both payers as well as the regulatory agencies will ask, and that at its very heart is the context of use. So every once in a while teach a class, thanks to Veronica at University of California, Berkeley, and at Stanford, and every once in a while I'll have a graduate student come up to me and say, why should I even be concerned about reimbursement? Why should I be concerned about regulation? And this is a slide I've started using. First of all, even though diagnostics only makes up about 4% of the healthcare expenditure, 4 cents on a dollar in the US, it actually informs how 50% is spent, about 25% on drugs and about 25% in terms of hospitals. So it provides a disproportionate value in terms of the money that we're spending. In addition to that, it's really important for us to translate this information as soon as we can sufficiently. I'll be returning to that, but we owe it to the patients we serve to get that information to them sooner rather than later. We've already talked about the accelerated translation, both on the standpoint of exploratory walks as well as directed paths. And then in addition to that, it's really critical important to have high levels of evidence. And but before I leave this slide, I also wanted to remind people who sometimes think, well, it's a diagnostic, it has to be cheap. Well, if somebody makes a commitment to gathering the evidence, a trial that we just completed and reported in the literature was a $15 million trial, and it was a diagnostic observational trial with 400 patients. If somebody offers you a cheap diagnostic, they don't have sufficient evidence to support its use. This next slide reminds us of the two paths for regulatory oversight. On the left is the CLIA, the service route. On the right is the FDA route. Before I leave this slide, let me emphasize that the FDA and the regulatory agencies are to be applauded for the way they've embraced change and the evolution of regulatory science in general, because in fact, a small number of laboratories have received regulatory approval, even though they're single laboratory tests. Robert, in fact, mentioned the AlloMap test, which is a test we have. There's only a handful of those laboratories, but it's possible. We've already commented on this. It's not inexpensive. Just to give you an idea, just to submit a PMA for diagnostics, just to submit is $250,000. You have to be pretty sure that you know what you're doing and you know how it's going to perform before you do that. It takes years to make the commitment going forward. This next slide is a reminder for how to think about analytical validity, and clinical utility. And across the top in the columns is the CLIA laboratory, the CLIA CAP, the College of American Pathologists. CLIA laboratories come in different flavors. But in addition to that, it could be a CLIA CAP laboratory that, in fact, has submitted their test to New York Department of Health to get the test approved, separate and distinct from the laboratory. Then you have the two familiar columns from the FDA, and you have the payer on the right. And one of the things that you'll note is that clinical utility, although required for a predictive FDA test and required for the payer, is actually not required for any other test. The payer is not interested. If you only demonstrate clinical validity, it's not sufficient for reimbursement. And in addition to that, comparative effectiveness health econ studies are key for the payer as well. So even though we have a multi-path regulated system, reimbursement alone actually determines what diagnostic tests are used. We have diagnostic tests that are FDA approved or not reimbursed. We have tests that are not FDA approved that are reimbursed. I'm particularly excited about Hans Eichler, European Medicines Agency, working closely with Janet Woodcock and her colleagues, now beginning to think about the levels of evidence from the standpoint of the benefit risk that patients bring to the table. In the past, we've thought about evidence is sufficient to treat everybody or insufficient to treat everybody, yet patients bring different benefit-risk ratios to the table. And Eichler and his colleagues and collaborators' papers point out that it should be possible in an adaptive way to allow a test or a drug to be used for a subset of patients rather than all the patients going forward. I don't have to spend any time on this slide. Robert, I think, did a nice job of describing for you that there are multiple questions in the context of a drug trial that a diagnostic can play a role. This just reminds you that if you were curious about developing a colinear development strategy with a drug and a diagnostic, there is a path to do that. There's a guidance to do that. Unfortunately, there are times when biomarkers come out of the wall and they have to be bolted on to these programs going forward, which makes it more complex. Again, thanks to the FDA's talks and the panel's discussions, I don't want to spend too much time with this, but I did want to applaud the FDA for having 30 diagnostic assays that are paired with drugs. That's with 17 drugs since 2014. 28 of those are companions. The label actually includes the diagnostic test, not the manufacturer, but the diagnostic test. And 26 of those are kits, whereas 2 of those, more recently over the last couple of years, are actually single laboratory CLIA-approved PMAs in the area of cancer. And there are 2 complementary assays. I think Robert did a nice job describing for you the distinction between companion and complementary. Complementary is fascinating because it's new, probably only 18 months old. The major distinction on this slide is that a companion diagnostic directs the practice of medicine, whereas a complementary assay informs the practice of medicine. Complementary assay actually doesn't appear on the label. I think biotech and pharma is going to find complementary really fascinating for a bunch of different reasons, not the least of which being because it doesn't narrow the number of patients who can use a drug. It simply helps those patients be managed. This is a slide that I don't have much time to talk about, but I'm more than happy to talk about it another time. It really presents what might be called an LDT, a laboratory-developed test bridging strategy, where a pharmaceutical company, a biotech company, can first offer the test through a service laboratory and then build a bridge to an in vitro diagnostic kit. I fortunately had the opportunity to both be at Celera, where we spent some time doing this, as well as at Quest, and I can't say enough about this strategy to minimize risk and contain costs as you're developing that. Things don't happen overnight. Some people think, well, if it's reimbursed, all of a sudden you're making tens of millions of dollars. This slide is a slide I put together when I first joined CareDx, percent covered lives, and tests sold on the left and right axes, and time across the bottom. And what you can see are key events that are indicated in arrows. And the take-home message, this is that AlloMap test that Robert and I mentioned. It's a gene expression test that's been offered since 2005. It got FDA clearance in 2010, 2008, and in 2010 it was incorporated in the guidelines. And one of the things that you'll note from that blue line, the test sold, is what drove it was the New England Journal of Medicine image, non-inferiority trial, and the incorporation into the International Society of Heart and Lung Transplant. So what are the take-home messages? Even though there's a multi-path regulated system, reimbursement really controls what tests are going to be used. The specific definition of clinical utility actually varies among stakeholders. They walk around the elephant and imagine what clinical utility is. A timely diagnostic, regulatory approval and reimbursement really requires a directed path. We know how to do that. We don't have to wonder. And then lastly, failure to reimburse appropriately will stifle precision medicine and translational science, whether it is an investment by NIH, an investment by NCI, or an investment by private industry. What does the future hold for regulated diagnostics? Diagnostics really determines how most of the healthcare dollar gets spent and is the keystone of precision medicine. Diagnostic test development is much more rigorous than in the past and provides an investment that's substantially increased over time. Neither CLIA nor IVD regulatory pathways satisfactorily address clinical needs. I will cut to the chase and say that just the suggestion that the FDA was interested in overseeing laboratory-developed tests has fundamentally changed two things. One is the FDA is now much more understanding and familiar with how rapidly tests have to be reviewed, how rapidly upgraded versions need to be looked at. And then secondly, I think that the CLIA setting, some of the laboratories in the CLIA setting have begun to understand that they have to up the bar in terms of the kinds of things that they're doing going forward. And lastly, payers don't have the ability to look at insight for all these diseases and increasingly look at professional organizations. That's one thing the NASH community can do. And then lastly, let me just say that it's important to consider NASH not as a single entity, as Arun and I were saying earlier, but what are the biomarkers for the NASHs? We need to distinguish between the equally valuable but different contributions of discovery and translation. We want to puzzle through the performance evaluation when the reference method is inaccurate. It's important to utilize the clear phased path for clinical grade diagnostic development. Value reimbursement of diagnostic tests is vital to test utilization. There's an evolving landscape of evidence and the use of adaptive licensing to balance access and safety. There's an exciting and evolving regulatory landscape that's unfolding that I have a lot of confidence in. And then the last 2 points about the NASH biomarker community, they're to be applauded for the biobank and registry efforts of Nimble, Litmus, Target NASH, and the Liver Forum's efforts to bolt together placebo arms of various trials for natural history information. And then lastly, it shouldn't surprise you, I think the NASH biomarker community would benefit from increased input from diagnostic perspective. Seems like there's a lot of advice from the drug side, but perhaps more from the diagnostic side. And then I'll just close with a tagline from the College of American Pathologists. Thanks to all the patients who participated in randomized controlled trials, subjects who participated in observational studies in the past, those in the future who provide evidence for healthcare decisions. Every number is a life.